Hello, I'm Meredith Blackwell, and I'm here at the International Mycological Congress 12 on the 12th of August 2024, and I'm going to talk to Teun Boekhout, whom I've known for a number of years, and he's going to tell us about his life story and his work in mycology. And Teun, if you want to go ahead and start, where were you born?
Well, I was born in a small village in the center of the Netherlands, called Hoevelaken. It is quite well known because of the traffic jams, already for ages. So it's a real farmland. You know, my grandfather had a small farm, but my father refused to work in the farm, so he became an optician. So middle class family, and then went to secondary school and became interested in biology because of my biology teacher. So he took us out in the forests, and that's how I became interested in field biology. And then initially birds mainly, later a bit of plants, and then of course mushrooms. And then the life of me as a mycologist started, basically. So I never lost my appetite for mushrooms. And now I eat them, really, I must say. I collect them. It's a little bit controversial issue in my country, in the Netherlands. But and then I started studying biology in Utrecht. And then for my master studies, I did a lot of mycology, actually. So initially I started lichenology with Harry Sipman, who is still in Berlin, retired. I studied lichens from Colombia, from the superparamos, high altitude Andean ecosystems. And then I moved to Leiden, actually, to the formerly called Rijksherbarium, now Naturalis Museum, to study a group of mushrooms in the Netherlands under guidance of Kees Bas, a famous agroecologist specializing in Amanita, and a real good mentor, I will say. Actually, I'm about to describe a genus after him. It's an Ascomycetes yeast genus, but anyway, I thought I need to give him a genus. So that will be Bas Ozyma. And then a group of people took me to Colombia to do fieldwork in the Central Cordillera, also high in the mountains, base camp 4,000 meters. And my job was to collect lichens and mushrooms. And of course, you can imagine if you collect mushrooms, you have to preserve them already in the field. But before drying them, you have to describe them, photograph them. And that in really the high mountain where we were was not so easy. It was a very hard job, but I liked it a lot. And at that time, my ambition was to become a tropical mycologist. That didn't happen really, although later on with, for instance, Aida Vasco, she was my PhD student from Colombia, I could do some work indirectly in the tropics. And but then I also started, I thought, for my masters I did already lichenology, mushroom work, ecology with mushrooms. I thought, let's do something different. So then I also did electron microscopy, but again with yeasts. And then I became interested in the Centraal Bureau for Schimmelcultuur.
I was wondering when you were going to get to the yeast.
Yes,
that's what I know you
exactly.
for.
So then I started studying for another small part of my master the ultrastructure of Ascomycetes yeasts with Sieber de Hoog, actually. And after my masters, I went back to Leiden to work on the Dutch agaric flora. They call it flora. It's not a flora, of course, but anyway, it's how it's called.
Yep.
And I worked there for 1 year, different genera. But I also knew that when there would become a vacancy available, I was not the first candidate of choice. Yeah, that's how you feel it sometimes. So then meanwhile, Sieber de Hoog wrote a project for a PhD student, and then I was the candidate for that between Utrecht University electron microscopy and the Westerdijk. And then in '87 there was a vacancy in the yeast department in Delft for Basidiomycetes yeasts, and I was invited for that job actually. And then I moved to Delft, entering the yeast division. So I did my PhD on ballistospore forming yeasts, basically, and then I still continue my research. I had to find my own research line, and then I switched a bit to clinically important yeasts like Cryptococcus and Trigosporon and Malassezia, and later on also Candida and so on and so forth. Doing mainly taxonomy, biodiversity, but I also had a strong interest in functional aspects. And one of the things that I also find very interesting from the day one that I entered the electron microscopy field was the ultrastructure of Basidiomycetes, particularly the septal cap. So we could also organize a project on that. We had 2 PhD students that working on them. It was really excellent, but the funding thereafter was also complicated. And I still think that is, if I could have followed that route on trying to understand that organelle, that would be great. But now I have to wait for young people to pick it up again, you know.
Yeah, we were talking about electron microscopy with someone else a little bit earlier, and I regret that people aren't doing more electron microscopy.
Yeah.
But I think it's coming back a little bit.
I think microscopy made such a huge advancement over time.
And now we don't have to use it to find characters because we've got the DNA, and so we can look as we like. Yeah. So you're working on Basidiomycetes yeast, and you're also doing other things. You're being a manager of a big project.
You mean the yeast?
Yes, I mean the yeast, what we call the Bible.
The Bible of yeast, yeah. Mm-hmm. Well, this was of course a very ambitious book series, starting also in Delft, by the way, a long time ago, maybe 80 years ago. And I was asked by Cleet Kutzman, the late Cleet Kutzman, and Jack Fell, who is still very active, by the way, 92, 93, still writing papers and still very, very, very much active, as an editor for the 5th edition. And that was quite ambitious, actually, 3 volumes, over 2,000 pages, 40 authors or so, or more even. I think 70 authors from 40 countries, something like that. And managing that was quite hard, you know. We went over that line.
Well, I remember talking to Cleet at one point, telling me he'd just finished proofing the index. And it was the length of more than a book. You know, it was several hundred pages for the index.
The index was hard, but it's still very useful.
Amazing.
Yeah. But now, of course, with Google searches, an index is not needed anymore.
So you were the young guy brought in to
at that time I was the young guy, and now I'm the old guy.
Yeah. So what big changes are you making?
That I make, or that I saw in my field?
Or that people are making in the Bible?
Well, of course, it's all now genomics.
Yeah.
I mean, of course, when I went to the US, I spent some time after my graduation in the lab of Cleet and Jack, just to learn PCR. I mean, that was a status. So just to do manual sequencing, you know, with these gels and technical
Very
problems.
difficult,
Reading gels backwards and stuff like that. I mean, it was fun, but it was, if you compare it now with what the technology development, it's amazing. Mm-hmm. And then, of course, the barcodes, ITS and D1, D2, developed initially by Cleet for yeast, Jack Fell for Basidiomycetes yeast. I mean, that was the standard, the gold standard, actually.
Well, I guess it was actually 26S for the yeast, for the Ascomycetes yeast.
26?
26S instead of the 18.
Exactly, it's the D1, D2 domains of the 26, yes. Yeah. And then ITS later on, of course.
Yeah.
And then, of course, alternative genes came up, and then, of course, the barcode paper, and now it's genomes. I mean...
Yeah, but every yeast now that's known has got a barcode.
Has a barcode, absolutely, more barcodes.
And so that's the first group.
And I guess that most of the Ascomycetes yeast now also have a genome.
Yeah, so that's just an amazing thing to have happened. There's no way I could have worked with yeast without having you guys who came before me with, the DNA.
Yes.
It's just no way I could have done it.
Yeah.
And Cleet gave me his whole database,
Yeah.
so I could look for insect yeast.
Cleet was a very generous man, I must say.
He was.
I appreciate it very much. I could spend some time in his lab.
Yeah,
And I still regret very much that he's not any longer amongst
that
us.
was such a surprise.
Yeah.
Yeah, about a month before he died, I was talking to him on the phone. He says, "Guess where I spent the night?" I said, "What are you talking about?" "In the hospital."
Mm-hmm.
And he had just passed out while cooking dinner.
Mm-hmm.
And so he was assumed to be okay, and then later I just couldn't believe it.
Yeah.
So anyway, very sad. But he was remarkable because I was involved with a group of people. We were looking at it was a Joint Genomics Institute group, and we were looking at some yeast and economically important ones outside of Saccharomyces.
Yeah.
Cerevisiae, and on the top of his head, he just could tell you the characters of all of these yeast.
Yeah, he had it all in his mind.
He had it all in his head.
Yeah.
The entire big table.
Yeah.
Yeah, amazing.
But it's not nowadays. It's almost all about DNA, and what we see, of course, in species descriptions, that people don't appreciate morphology. People don't appreciate the physiology. I think it's still very useful to understand the biology, you know.
Yeah.
And I think just reducing a species to a sequence, you know, I see the discussion in the eDNA world. I understand that completely. But if you have a species real in hand, you should give a description with relevant properties, I think.
Yeah, and I noticed that we would work very hard. It was very difficult in our lab to do all of the I guess we'd end up with 200 characters for the yeast. There were other, well, some of them were morphology, but most weren't. But just to keep all of the media going and have them ready to do it. It was really difficult work, and it should be done.
Yeah.
People shouldn't stop
doing it. But I see it ending, you know.
More, yeah. Yeah, and people stopped doing some of the fermentations.
Yeah.
You know, I
also,
noticed
I see that many people don't appreciate the microscopic morphology.
Yeah.
You know, and I think even with a so-called simple organism as yeast, you can still see a lot. You know, both colony-wise, but also in the microscopy, the shell shape, the budding.
And I think it's amazing that people got so much right about the yeast before there was DNA. You know, it's amazing to
exactly.
me.
Yeah.
I could never have done that. And there's no way without the DNA, though, that we could work very easily.
So now it's all the whole genome stuff, and I think that's exciting. I mean, particularly if you see the technological developments that you now have kind of an USB stick on your computer where you can generate your DNA sequence, and overnight you have your whole genome stuff done. And of course you need computer skills to deal with the data.
So how many genera are you going, do you have any idea how many genera were in the last edition of?
I don't know exactly, but this morning we had a discussion about the yeast.org, and I mentioned 300 yeast genera, 170 Basidiomycetes, 130 Escos. Vincent Robert corrected me saying 270. But I think he might be outdated, or I might be too optimistic that already I include genera that are on the way.
Yeah.
Of course, the big problem, or whatever you call it, to be solved is Candida. Candida is all over the place in the Saccharomyces, and that is not sustainable, so to say. So we are now working on a paper with whole genome data, different parameters to propose a solution for Candida.
So the Candida will be done before the Bible.
Yeah, but I think there will not be a book anymore. I think that the idea for the yeast.org is to have an electronic database.
Yeah. Oh yeah, definitely.
You know, the website, the yeast.org.
Yeah.
And of course, if people want a printed version, that can be done, but then there should be sufficient demand, of course. But of course, you can more easily update an electronic database than a book. You see,
Oh yeah,
but,
and as you say, you don't need an index. It
and you don't need an
takes you a
index. You
year
simply
to finish.
exactly.
Yeah.
So,
Yeah.
and we hope that the community, because I think this is a community effort. That the book has already been a community effort over the years, but also this is a community
Be more
effort. And likely next year, 2025 already, maybe we should have a 2-day meeting just on this project with the stakeholders, the participants, to see how to better organize it.
Yeah.
And also for me to hand over, because that's also what is done.
You got to
what is
find
needed.
another youngster to take it on.
There is I think there are some people, but you need to transfer things in time,
Yeah, yeah.
So that is.
It's a big job.
It's a big job, but I like it.
Yeah,
And I also like the community that is becoming younger and older, of course.
and you're in a position where you get credit for, well, before you retired, you get credit for doing what you're doing.
Yeah.
I think that's sometimes a problem with academics in the United States anywhere, where you've got a dean who wants you to bring in money, grant money. And so that sometimes is a consideration.
I do think, though, that nowadays, so when I entered the field as a young scientist quite some time ago, I worked for 40 years at the Westerdijk, so then that's the time scale.
Whoa.
Well,
I had no idea it was that long.
I think the life was easy then. I mean, in fact, there was no need for grant writing. You had your salary. There was a bit of money for consumables and chemicals. But nowadays that's completely different.
Yeah.
So there is a permanent rush for money, and publications, and so on and so forth.
Yeah, I always felt I had complete freedom and I could work on weird fungi that nobody else was interested.
I also experienced.
Yeah. But some of your work on the Dollypore septum, I just love that stuff.
I loved it also, but I couldn't. For that, I needed external money.
Yeah.
And that was hard to find. I thought it is interesting because it was a novel organelle, basically. And of course, if this was in an animal cell, I mean, this was all over the news. But it was in a fungal cell, and then they said bye-bye. But it's still amazing, and I still am absolutely sure that one day somebody will pick it up again and find very amazing things.
Well, I think that I've lived at a really good time, some of the best of times for mycology, because we were pre-DNA and post-DNA, but we also got the EM part, and so it's
but it's
really
of course
been
difficult
fun.
to predict the future in terms of technological developments, because when I started microscopy, I thought that is the end of microscopy. I mean, that the microscope at that time was the end of development. That turned out completely wrong. Same with DNA. I mean, I started manual gels and whatever, and now it looks like easy.
I went out to Berkeley to learn how to do PCR, and I can remember my first gel. It was all hand loaded.
Exactly.
And Tom Bruns and I loaded stuff on the same gel, and we did it just the opposite. So it made a beautiful pattern. It was like a Rorschach diagram. If we folded it. But I was so excited. I took the gel home to the hotel where I was staying, and there was a little lamp, and I held it up, and I read the gel by this lamp. And it turned out it just didn't make sense at all. But I was reading from the wrong side.
Exactly. I mean, mistakes happened. I also made many mistakes, by the way.
So anyway, it was really exciting.
There is no progress without making mistakes.
Oh, no.
That's how I feel it, and others can make corrections. I can make my own corrections, but without mistakes, no progress.
Well, things like in your Basidiomycete yeast, the genera have changed so that they're more natural.
Yes, and I was so surprised because, well, that was a major taxonomic change. And before publication, I thought, well, they will kill me all because of this proposal. And of course, I was not the only author of this work, but I think I was really kind of steering it. But overnight, it was accepted. And I think because it made a lot of sense in the end. And of course, you can still discuss things, maybe some genera are still wrongly circumscribed or too small or whatever. I mean, that is ongoing field.
It gets refined.
Exactly, it gets refined, and you also have to leave something for the future for the young people.
But, you know, and people start looking in different places and they find completely new yeast.
Wow, look at your own work. I mean, all your insects, I mean, it's amazing.
Yeah.
You know, I mean, I still think that was that Candida tanzaniensis clade went from one species to, I don't know, and now it's a genus.
And you know, that was Sung Su, who was in my lab. He came for 6 weeks. Professor Sugiyama, I said, can he come, because he's been working from culture collections, and I want him to do a little field work. And so we had some people working in the lab on labules. And so we decided, well, they're working on labules, we'll look at the insects that they're catching to get the labules off of and see what's there. And so Sue worked on all of the protocols and got them going so that we were finding all these things. And he came and told me, and at the time I had no idea what Candida tanzaniensis was.
Exactly.
You know, it's from some lake in Japan or something.
Yeah, just one strain maybe.
Huh?
Maybe there was only one strain at that time.
That's all it was. And so he said, we're getting some more like this. And it took a minute for me to realize that this was really unusual.
But another, I think, very nice example is Spathospora.
Yeah.
I mean, also going from one species to...
Yeah.
And have also technological implications.
And that was another insect associated one. And that was an undergraduate. You've met Sung Su. I mean, you've met Nu Nguyen.
This morning.
And Nu was an undergraduate, and he kept working and working and working on it so that he could get the ascospores to develop. It was amazing. And now there are, how many species do you think?
Oh, for Spathospora, I don't recall actually.
And Spathospora
more than 10, I think. 20 maybe. Maybe 20 species or so?
Yeah, there are a number of them. Yeah. And they become really important for people that are trying to ferment other sugars.
And actually I'm collaborating with other people following up your insect work. Because I think when there is interesting species of beetles in Louisiana, there must be interesting yeast in insects in Botswana. And with Nerv Zhao, a young guy in Botswana, he's a very prolific writer, by the way. I think he will make a good career. We find lots of new species in Botswana.
Well, if you look at the ones in the mushroom-eating insects, those are really interesting, and we're finding some of the same ones in Australia and are close. You know, they're all close.
And what I think is also interesting when you look in the Candida albicans clade, so the Lodoromyces clade, where most of the important Candida pathogens are, almost all the species from that clade are from insects. So I had the idea, and it's difficult to test, that maybe the insect habitat prime those yeasts to avoid the human immune system. It's a theory. I told it to Nerv and said, well, try to prove it, or disprove it, of course.
Well, the other thing that I wonder about, and I'm not quite convinced about how some of this is happening, but if we're looking at some of these in the same habitat, general habitat, like a beetle, and we're looking at this in different places, you know, in Asia and Australia and the U.S. and Central America where we've looked, and you find they're all in the same clade. And so how have they spread and when? You know?
And what are they doing with the insect?
And I think that in some cases, some of the mushroom-feeding, some of the wood-eating beetles, I think the yeast are just there using the habitat. And they get what's left over that the beetle hasn't digested.
So exciting environments, lots to do for the future, for the young people.
And the worst part will be done because the taxonomy and the species will be discovered. And that's the hard work.
That also will never be finished.
No. Okay, Teun, anything else to say about any of this?
I really appreciated to be a mycologist. I still like it to be a mycologist. Both, you know, scientific-wise, but also cultural-wise, you know. I enjoy beers, I enjoy wines, I enjoy edible mushrooms, so that's a great part of my life. all thanks to the fungi.
All enhanced by fungi. Yeah.
And thank you for inviting me.
Oh, yes, definitely. Thank you.
Okay, thank you.